Hello, everybody. This is a module of the OpenCAPI course on accelerating big data analytics applications with FPGAs. This module will focus on the acceleration of genomics algorithms, genomics being one of the most important big data analytics applications in the field. The outcome of this module will be as follows. First of all, we will start with an introduction to genomics, describe the various different applications of genomics and its impact on society. And then we will discuss a number of different important genomics algorithms that are used in practice. Then we will discuss an acceleration on FPGAs of DNA mapping algorithms. And finally, we will end with an acceleration of variant calling algorithms. Both of these algorithms are some of the most computationally intensive algorithms in the genomics field. My name is Zaid Alars, and I will be your instructor for today. I am a professor in Delft University of Technology in the Netherlands, and my research focuses on building accelerated big data systems and integrating FPGAs into the big data ecosystem. Let's start this talk with a couple of definitions. The field of genomics is the field of the study of the genome. The genome itself is the organism's complete DNA information contained in all of its cells. It represents a program that defines the way the cell functions and how the cell is structured. The field of genomics represents an important and growing field that helps us understand disease, provide appropriate medicine, help us improve our agriculture and livestock, and also in understanding life as we know it. This field is a highly demanding big data application domain that requires the analysis of immense amounts of data using complex algorithms. DNA encodes information as a sequence of 4 molecules. These molecules are usually referred to by 4 letters: A, C, G, and T. The molecules are also referred to as nucleotides or bases. And since the DNA is always represented as 2 strands of complementary bases, these nucleotides are also referred to as base pairs. A gene, on the other hand, is the smallest functional sequence of DNA. It encodes the way to construct proteins that have a specific function within the cell. Since the DNA defines the cell functionality and structure, changes in the DNA can result in failure of the cell to function. These changes to the DNA, or these mutations, can result from environmental factors like ultraviolet radiation, or from incorrect copying of the DNA by the cell. Most of the mutations that happen in the cells have little impact on the behavior. However, sometimes some mutation can prevent normal operation, which results in genetic disease, such as hereditary diseases or cancer. We are able to diagnose these genetic diseases by reading out the sequence of the DNA in the cells of an organism and comparing it with a reference DNA. This way we can identify the differences in the diseased cell and comparing these differences with the sequences of well-known diseases that have a genetic source. Reading the DNA sequence of an organism is referred to as DNA sequencing. DNA sequencing has traditionally been a very expensive operation. However, the price of DNA sequencing has dropped significantly in the past 10 to 20 years. This is the result of very fast technological developments in the field that resulted in reducing the price even faster than the exponential decrease dictated by Moore's law. As an example, the price of sequencing a human genome has dropped from somewhere around $100 million in the beginning of the 2000s all the way to just under $1,000 around 2020. That's a decrease of about 100,000-fold compared to the price 20 years ago. This trend is expected to continue as this field is being developed quite rapidly and multiple technologies are being developed continuously to reduce the price even further. As the price of DNA sequencing has decreased in the past years, the amount of DNA data that we have been collecting has been growing exponentially at the same time. Various different organizations created databases of DNA information that in many cases are shared publicly to increase their utilization. At the same time, we have been developing more and more complex analysis methods that help us mine this wealth of DNA information and allows us to have better understanding of the variations in the DNA. This has resulted in multiple challenges in genomics, both computational challenges in analyzing the data and also big data challenges in acquiring, storing, and managing the data that we are collecting. These challenges in data analysis and data collection prevent us from applying DNA analysis for a wider population. In order to make DNA analysis practically viable, we need to reduce the time it takes to perform the analysis and also reduce its cost. Reducing the compute time of DNA analysis allows us to apply it for urgent clinical diagnostics applications. For example, applying it for targeted cancer therapy, what is called precision medicine, or applying it for neonatal diagnostics cases. Reducing the cost of DNA analysis allows us to make it available for an increasing number of patients and also increase the number of diseases we can use to be able to diagnose with this approach. This will reduce the threshold of applying this method for a wider population and increase its benefit to society. One of the most widely used DNA sequencing approaches today is referred to as next-generation sequencing, or NGS. The process of next-generation sequencing starts by taking a sample from an organism and supplying the sample to a next-generation sequencing machine, for example, an Illumina high-seq machine. The NGS machine does not generate a single continuous strand of DNA sequence, but generates about 1 billion short reads of about 150 base pairs each. These reads are distributed randomly throughout the DNA, and these will have to be stitched all together to reconstruct the original DNA of the sample. This is done using various different analysis algorithms. The amount of data that the NGS machine generates amounts to about a terabyte of data every day, and the time it takes to process all this data in downstream analysis would take about a number of hours to be able to complete, depending on the analysis. Computational pipeline starts with a file that is generated by the NGS machine. This file is usually stored in the FASTQ format. The file contains the exact basis or sequence of every individual read, that short read that was generated by the machine, in addition to a quality parameter that defines how accurately this read has been generated. The FASTQ file is then mapped using a mapping algorithm to identify the location of each individual read in the file by comparing it to a reference DNA. The reference DNA represents the expected gold standard of the DNA of that specific organism. This way, all the short reads are aligned in their expected locations on the reference DNA. The output of this mapping mapping algorithm, is usually stored in the SAM format. Afterwards, the reads in the SAM file are sorted, are cleaned, and locally realigned so that a more accurate location for their position on the DNA can be identified. Finally, we use this readjusted file and compare it with a reference DNA again to identify the mutations or the variations in the sample DNA compared to a gold standard DNA. This process is called variant calling, and the output format of this process is a VCF file or variant calling format file. One of the most widely used variant calling pipelines is called the GATK pipeline, which refers to the Genome Analysis Toolkit. GATK has 5 main stages. It starts with the BWA algorithm, or the Burrows-Wheeler aligner algorithm, which maps the short reads stored in the FASTQ file generated by the sequencing machine to a reference genome to identify their positions. Then Picard sorts these mapped reads and marks duplicated ones to prevent using them in downstream analysis. Then the Ender realigner locally realigns these reads to identify a little bit more their accurate locations. Then the base recalibration algorithm recalibrates the quality parameters of the bases of each one of these reads. And finally, the haplotype caller is a variant caller algorithm that identifies the differences between the bases of the sample DNA compared to a reference DNA. In terms of computational challenges, 2 main stages of the GATK pipeline are particularly computationally intensive. First of all, the BWA algorithm, which represents the mapping stage of the pipeline, consumes around 30 to 40% compute time of the whole pipeline. And second, the haplotype caller, which identifies the variations or the mutations in the DNA, consumes anywhere between 40 to 50% of the whole compute pipeline. In both cases, the BWA and the haplotype caller, the time-intensive function can be accelerated effectively using systolic arrays on FPGAs. For the BWA algorithm, we accelerate the seed extend part of the algorithm that can be done in on FPGA. And for the haplotype caller, we accelerate the pair HMM algorithm. We will discuss these different algorithms later in the module in more details. There are a number of ways that we can use to increase the performance of computationally intensive functions in the genomics pipeline. One approach is to increase the scalability of these pipelines and allow them to run in parallel on multiple nodes in a cluster environment. This way, we are able to reduce the user time of these pipelines. Another approach is to accelerate computationally intensive functions of these pipelines on a single node. This way, we are able to reduce the actual compute time of computationally intensive functions. In the rest of this module, we will present 3 different ways to reduce the runtime of the GATK pipeline using these 2 approaches. First of all, we'll present a big data scalability framework that allows us to run the whole GATK pipeline in parallel in a cluster environment. Second, we will show how to accelerate the seed extend algorithm of the BWA mapping program on FPGAs. And finally, we will show how to accelerate the pair HMM algorithm, which is part of the GATK haplotype caller on FPGAs.